Hello to everyone. This is the Bespoke Gene Therapy Consortium seminar, and we are planning to discuss two RSP. People in the panels are Courtney Silverton, that is in charge of the program manager for the entire project, Brad Garrison, that is the project manager, and he will answer questions about either responsiveness or compliance of the applications. And Yang-Hei Su, that she's an expert in the AAV biology. She will answer the questions related to the biology. And myself, I'll do a short presentation of each one of the RSPs. And my function here is the co-chair in the AAV biology team. So we are presenting two different RSP. One that is enhancing the therapeutic gene expression in human gene therapy, and the other one is enhancing the AAV production for human gene therapy. You can go to the next slide. So it's very important that when you apply, you mention clearly what is the RSP you are referring to. And two important days. One important date is that the deadline to receive any application is by midnight of October 28th Eastern time. If I write later, the applications will not be accepted. So now in the next slide, what we want to do here is to explain each one of the RSPs. Both of them, the idea in the RSP is to increase the efficiency on the production and in the genes. So the first one is enhancing therapeutic gene expression, and you can read, but the overall goal is to advance the understanding and how AAV transduces tissues, then develop approaches to improve the transduction, and the proposal can be focused in any of the key mechanistic steps involved in the transduction. Examples in this case are extracellular transport across the BBB or extracellular transport through different tissues. In the next slide, the specific research objective here is to increase the fundamental knowledge about AAV transduction biology and hopefully develop solutions for this. In the next one is the specific areas of research in this particular RSP. I will read just part of it, but you can read, this is directly from the RSP. So optimization in any of the different mechanistic steps that are highlighted in the slide. These steps are not, it doesn't mean that you have to pick one of these steps. You can take one or more, and it may not be in the list. If whatever you propose is not in this list, please contact Brad to see if whatever you plan to do is responsive to this RSP. In the next one, the more important is which applications are not responsive. Areas of research that we are not interested in this application. So one is effort to improve hepatocyte transduction efficiency. When whatever you do has to be not specific to a disease, but a general one. And the research focus on host adaptive immune response. That is autoscope to this application. And finally, gene editing is also autoscope. And the last one in this RSP, the next slide, is, okay, how do we do the review? The review is first merit and rigor, but will also include other review criteria, and this is also in the RSP. So is first, the work proposed is focusing on investigating and improving mechanistic steps in AND transduction, either extracellular or intracellular. Is the work likely to result in clinical relevant enhancement of therapeutic gene expression in patients? This is really important. Is the work addressing a high-value clinical question for human therapy? Will the research outcomes or technologies developed be broadly applicable for human gene therapy across disease indications and cell types? Now, NCATS is one of the participating and the lead institute or IC in NIH, and that is the project proposed NCATS HTS capability and are compatible with the screening platform. This review criteria is not exclusion. If you are not doing this, still they might be acceptable to us. And then is, can you do the work in the time that you proposed? It will be important that you, in your write-up, you answer, not necessarily each question, but you address in the wording these questions. Now, in the other RSP, that is for increasing, so next slide, I'm sorry. So this is the other RSP we are changing. Now we are in the enhancing AAV production, vector production, different RSP, different intent. So here is to advance the understanding of how to improve the production of AAV. Also how to understand the AAV viral cycle and the molecular steps, and we are involved in the generation of the recombinant AAV vectors. The idea here is leading to improvement on the vectors manufacturing. So in the next, very similar to what we did in the RSP VGTC 003, the technical objectives here are to develop or investigate solutions to improve one of the more steps in the recombinant AAV vector production. And ideally, it will be applicable to any AAV serotype. The broader is better. In the next slide, the specific areas of research, again, here I will let you read, and if your area of research is not in this list, doesn't mean that it's not interesting to us. Please send an email to Brad to discuss it with the team. Now, more important is the next one, the next slide, and where the areas that we are not interested. So this one, I will paraphrase all of them. So research focus on AAV processes, development improvement, scale up and purification is not for this RSP. This will be it will be considered not responsive. Effort to increase the AAV therapeutic gene expression, that is not for this RSP. This is for the other one. And don't assume that you send it to this one and the people that receive it will automatically transfer it to the right one. That is not the case here. We cannot guess what the PI has sent it. You submit it to this one, it will evaluate it as an application for this RFP, not for the other one. So make certain that you clearly pick the RSP that you want. So research focus on the host in adaptive immune system is out of scope, and the same, gene editing will be out of scope. In the next one, sorry, is the review. So what we are going to look at, like in the previous one, besides merit and rigor, it will be, these questions are, is the work likely to result in substantially enhancing the efficiency of production of the AAV vectors? It will be how big will be the enhancement tool will be really important. Is the approach scalable? Potentially compatible with the use of a GMP facility? Again, really critical question. Will the research outcomes or technology will broadly applicable for manufacturing human gene therapy across AAV serotypes? This again is the broader, the better for us. And the next two ones is exactly the same that in the previous RSP. If you use the NCATS HTS facility, it will be okay, but if you are not using it, it doesn't mean that it's not acceptable to this RFP. And can you do it in the time that you proposed? So the next one is for questions. So in the way that we are going to answer questions is we'll do it from the chat, but I think first Brad has a few the question that we received before that he will address first.
Sure, thanks, Enrique. Before we get to the questions, if we could go back to slide 12. So just wanted to highlight that for both of these RFPs, we've listed some items that are considered non-responsive or out of scope. If you actually go into the RFPs themselves, some of these out of scope items, we actually have links to other non-FNIH funding opportunities. So there's an accelerating research and development for advanced therapies, which deals more with host adaptive immune responses. And there's a PAVE GT program, which is more for gene editing. So specific links to those other opportunities are in the text of the RFP, if you find the scope section. So we received a question in the chat asking if the solicitations have been announced. And indeed, they have been. They've been live for a good couple of weeks. And the link to the opportunities page is there in the chat. So just getting to some of the questions we received, got an email, some questions sent by email. The first question is, are there institutions responding to the RFP for improving expression that would like to partner with us? So in general, we certainly can offer, connecting organizations together who are interested in collaborating. The submitters are responsible for identifying any third-party IP needs, including from BGTC research partners, which refers to other awardees. We have no way of knowing who the research partners will be until the awards are made. Now, for private sector companies, they might be able to identify and propose research elements that could advance their own pre-existing IP or pipeline without needing IP protection on the results themselves. And they're welcome to propose such research and utilize it for their own internal purposes, as well as making it publicly available. So there were actually two questions. So one was about collaboration and one was about the pre-existing IP considerations. So I hope that helps.
I know as we're waiting for additional questions, maybe I can also ask a question on behalf of some of the academics on the call. You know, I was an academic for many years before I transitioned to industry. So I think if I was an academic thinking about applying for this, I would wonder, number one, like, what is the success rate? Right? You know, I think the last time I applied for NIH funding, not FNIH, but regular NIH funding, the chance of actually getting your application funded was like single digit. It was very, you know, it was very hard. So is there anything that we can share with the audience here in terms of what are the chances of having an application successfully funded?
Oh, that's a great question, John Hay. This is actually the second round of RFPs on these topics. In the first round, we received 13 applications and ultimately accepted 4 of them for funding. So approximately a third of the applications received were accepted. That being said, we're anticipating about 7 more awards. So there clearly is an opportunity there for the folks on the call. You know, I see we have 17 attendees. So if that's reflective of the applications we receive, I would think your odds would be pretty high. I think the real key is to focus on making sure that you meet the scope requirements and avoid some of the items noted as out of scope.
Yeah, that success rate is pretty remarkable, right? So I hope people took note of that. I think the other question I would have, again, asking on behalf of the academics on the call, like, how is the review process similar or different from regular NIH review process, right? So this is a different mechanism. So again, how is it similar or different from regular NIH review? And
Well,
maybe, I don't know. Oh, yeah, and Brad and Enrique. Yes, please.
Yeah, so well, we at the FNIH have established our own review process. It's patterned after the NIH process, but it is not exactly the same. So we assemble a panel of experts to do the reviews. They're done in triplicate. There's a scoring criteria that is taken into account, which is listed in the RFP. And so that's how the reviews are done. Then after the primary review, we take it to the sub-team for a secondary review, and then ultimately the final decision is made by the BGTC steering committee.
To reemphasize, is first in the first round, let's call it the success rate was higher than single digit. So substantially higher, I would say. Second, the review here is peer review, but are people that are from industry and academia, and the focus is in the quality. So getting approval or not will depend on what is how the application responds to the RFP, and second, what you are proposing has really merit. But the success rate is certainly no less than 10%. It's substantially higher than that. And I would like something, maybe it's my mistake, there is a question about deadline. It's August. Maybe my accent was misleading, but it's August, no October. And the RSP is right there.
Yeah, August 28th.
Yeah, and I would just wanted to add around the review process. It is a lot more agile, I would say, than the more traditional NIH review process. The Bespoke Consortium, we are able to make adjustments on the fly of how we do this, right? Based on our needs. And so, please, if you have any questions or concerns or anything, you just wanted to float by, reach out to Brad. We can even have pre-submission calls just to see if what you are thinking of proposing would be of interest to the consortium, right? So we can give some general feedback as well. We are really interested in getting the highest quality proposals possible. This is a really important area to really increase the knowledge around AAV biology, both the transduction as well as the production side. So we are happy to work with you to ensure the highest quality proposals. Okay.
And you will have the opportunity to answer the reviewer critiques before final decision, unless there are no responsiveness problems or some critical issue. But if not, you will be able to answer the critiques before we make the final, the steering committee make the final decision.
And we've received a few questions in the chat. One from Anna Cajaste out of Italy. Are research teams outside the US eligible?
It was answered by Courtney already, yes, and I'm sorry to use this word, foreign institutions are allowed to apply. I don't know if you want to add something, Courtney. But it's already answered in the chat. We can go to the next one.
Yes, the only requirement is compliance with the AMP policy. So we welcome all organizations, US-based, international, for-profit, and nonprofit.
Okay.
And is there a form, and are there specific indications on how to build the proposal?
Yeah, well, yeah, those instructions are included in the RFP. There's a section called Proposal and Submission Instructions. So we have a 10-page limit for the primary response, and then some additional requirements for bio sketches and things like that. There is a submission form, which is Appendix 1 in the RFP, where you enter the title of your project, your name as the investigator, submitting organization, and that type of information. So
Can we show
the RFP?
Can we show that section of the RSP? Because that is there, right? So I don't know if we can show it now.
Yeah, let me pull it up. Let's see if I can take over the sharing. Okay.
And it's very similar to both of them, so with one is enough, right?
That's right. Yeah, they're both almost identical. So here's Appendix 1. This is the submission form. So this is where you can identify which RFP you're responding to. You can enter PI and any co-PI information, as well as total project budget. So when you're replying to the RFP, we've identified some sections here, right? Section 1 being the project overview with specific aims, deliverables, and a discussion of the applicability. Section 2 is the scientific design. So background, supporting data, experimental plan, analytical methods, technologies, assays, human and animal subjects. Section 3 is where you describe your data sharing and intellectual property management plan. Again, you're allowed to have pre-existing IP, but any new IP that's generated would be expected to be in the public domain. So here's where you can describe that information. Section 4, timeline, milestones, deliverables, and budget. I think that's all pretty straightforward. And then we have a budget table. So these are 2-year awards. And so you can fill out the budget table for the different costs in year 1, year 2, and the total. Section 5 is program support, listing key personnel involved in the project. And section 6 is any expected legal agreements, material transfers and things like that that might be relevant. Then for attaches, the budget justification, bio sketches, and any letters of support that you might have.
I think the other question I may have is around preliminary data, right? Is preliminary data required for this application? As you were going through it, I was, that's the question that came to my mind and something I would be wondering, because as we know, a lot of other NIH funding mechanisms, it almost sometimes feels like you have to have solved everything and done everything in order to apply. But, I think in the spirit of this RFP, we're really interested in having people who even sit outside of AAV field apply their expertise to solving the challenges in AAV, right? So people who may have very little experience with AAV, but maybe they have some really interesting expertise in synthetic biology, let's say, and then if they're able to apply those skills to investigating and solving some of our AAV specific, I think that would be really interesting to us. And so, maybe just answering my own question could be that you don't have to have, shown that you already done everything that you're about to propose. It's more like if you can convince the reviewers that you have the expertise, you have the ability, the capability to ask and answer the questions that you're proposing. I think that should be sufficient. So you don't have to have a long, lengthy CV in the AAV field to be eligible and to be regarded as a potential candidate.
And the next question, for the RFP for increasing vector production, should the focus be on upstream process manufacturing, since process development and purification and scale up are out of scope?
Yep, I think that's the right way to think about it. It is more upstream. It's about the producer cells, the plasmids that are used, whatever platform. Yeah, so upstream would be the right way to think about it.
And the next question, in terms of team composition, length of the proposal, are you looking for multi-PI teams?
Well, there's no requirement for multiple PIs, but certainly we would entertain that. The BGTC believes in a team science approach, so we're all about cross collaboration. So it would be great if you had multi-PIs, but it's not required.
Yeah, agree with that. And the other thing that we're trying to do with, for our awardees is to develop a network. So once people have been awarded funding, we want them to connect. And so we're thinking of ways to having them present their work and their data to other awardees. And so there could be kind of this cross-pollination on the other side of funding as well.
And just a quick reminder to those in the audience, if you would like to ask your question verbally, you are free to do so by using the raise hand function, and we will ask you to unmute. But the next question in the chat, does technology development for quantification of general AAV efficacy fit the criteria?
So that sounds like in vitro potency assay, things like that. I think that's the way I'm reading that, but feel free to correct my understanding if that's not it. I think we're looking for something a little bit more fundamental to the biology of AAV that could then lead to enhanced transduction or enhanced production. And I think this is a little bit more process, this particular topic that you're proposing. So I think it would be a little bit tough for this topic to get funded.
Okay, do any attendees need for their submission cell-based assays for complement activation, which is associated with AAV adverse events?
I think I'm hearing somebody volunteering services for complement activation assays. So certainly if any of the attendees, that's an area of interest. I think if you wanted to message Violet, that sounds really, really interesting.
Is safety of vector production platforms to host cells, e.g. integration, innate immune activation, within the scope of these RFA?
Safety of vector production to host cell. So I'm seeing integration as an example. So integration of the AAV vector in the target host cells. Okay, so I think I'm a little bit confused. So whoever asked this question, if you wanted to come online, because are we talking about the vector production RFP? Are we talking about the transduction part? Yes, please go ahead.
Yeah, can I speak? So my question is, if the production platform for example, insect cells or 293 cells, and then the subsequent, so what is produced with this, within the same serotype, etc., how that differentially affects the target cell, either by integrating more or less, or by activating the innate immune system of these target cells more or less. Is that within the scope?
Interesting. So you're thinking of either studying or manipulating the production platforms to then impact different vector properties, right?
Exactly. Yeah, like or at least compare the different production platforms that are there. I'm not into production platform development, but more in sort of testing the safety of the different existing production platforms that are out there, and then also the variables to the vector serotypes, etc. Would that fall within the RFA?
Yeah, so that would go towards enhancing the vector transduction, right? Even though you are using the vector platforms and you're evaluating different vector platforms, the ultimate goal is to understand how to enhance vector transduction. So I think my immediate read of your idea would be to go for the enhanced transduction RFP, and that could be interesting, right? To understand what is it about these different production platforms that lead to different vector properties that then impact transduction. Yeah.
Okay, thank you.
Will AAV dual vector development and enhancement of expression efficiency from such vectors fit the criteria for this RFP?
Yep, that is an area. We know that dual vectors, there is certainly interest in the field of using dual vectors, especially really large gene editing cargo, right? But again, we don't want this to be about the optimization of gene editing or the gene editing cargo per se, but if the focus is really around improving ways for dual vectors to get in and then do that recombination, either at the DNA, RNA, protein level, whatever, right? And then that approach can be then more generally applied to different types of cargo. Gene editing cargo could be one, but it's more broadly applicable. I think that could be interesting, certainly.
It looks like we've gotten through all the questions posted in the chat. Oh, here we got another one.
And Deepa asks, in that case where safety of production platform, we have both insect and mammalian production platforms, we can collaborate.
Yeah, there was actually a comment made during the review process in the first round that there would be an interest in seeing a comparison of different production platforms. So I think that's a good topic and worthy of consideration.
Okay, seems that there are no other questions. If anyone has any questions or further question, you can direct them to Brad, who will run directly internally. I hope every one of you apply. That is what's the reason of this webinar. And I don't know if anyone has anything to say, Yang Hei or Brad or Courtney.
Yeah, no, thank you so much for attending the webinar today. Really appreciate your time. Again, to emphasize again, we're really looking for high quality, high caliber applications. This is a really important area for the Bespoke Consortium. I hope you, I guess, I don't know if it's clear through the webinar, but things are done differently for this consortium compared to regular, more conventional NIH funding. So I really hope that you take advantage of this very unique opportunity to impact the future of AAV gene therapy. And we all look forward to your applications.
And one last question came into the chat. I see it's been answered, but yeah, the window to submit disease nominations for the clinical program has closed. That actually closed back in February of this year. So okay. Well, yeah. Looks like no more questions are coming in, so we can close the webinar a little bit early. We appreciate all of y'all taking some time out to attend, and look forward to seeing your applications.
Yeah, best luck in your submission.
Thank you, everyone.
Thank you, everyone.
Bye-bye.